Hi and good day everyone, I'm Nick Aludin Hafizin. This video is about effects of elevated CO2 on the morphological and physiological properties of C3 plants.
To briefly introduce the study, the atmospheric concentration CO2 has increased from its pre-industrial value of around 280 ppm to its present level of 400 ppm and is expected to reach 570 ppm by 2050. The key environmental elements that govern plants' growth and controlling the photosynthetic process is light intensity and CO2. The steady increase in atmospheric CO2 influences the overall physiology, growth and development of crops. The change in phenology, growth and energy of crop leads to change in quality of the produce. Plants do respond positively as well as negatively to the elevated levels of CO2 based on their photosynthetic pathway. Therefore, a better understanding on the effects of CO2 on C3 plants' growth is needed and timely to be investigated. IPCC recorded continuous global warming caused CO2 levels rise to 670 ppm by 2100. Generally, by influencing the photosynthesis process of C3 plants, the elevated CO2 can affect the yield, physiological and morphological of C3 plants, as well as fruit firmness and quality of C3 plants. Most crops, particularly the C3 plants such as rice, barley, soybean, and tomato, appear to react positively and negatively to elevated CO2 levels without the presence of other stressful factors. The importance of this study is we can know how C3 plants react to change in atmospheric CO2 and give insight on how elevated CO2 helps the C3 plants such as tomato and rice to increase their productivity and yield. The study also helps us to acknowledge the importance of balancing CO2 levels and give awareness to us. There are 5 research hypotheses that you can see there. There are also 5 research questions that you can see in this video. The objective of study is to investigate the effects of elevated CO2 on the photosynthetic and transpiration rates properties of the C3 plants, to study the effects of elevated CO2 on the morphological properties of the C3 plants, to determine the effects of elevated CO2 on the yield productions of C3 plants. Effects of elevated CO2 on the physiology of C3 plants. The photosynthetic rate of C3 plants rises by around 58% because of elevated CO2. Their reaction to elevated CO2 more favorable compared to C4 plants. Elevated CO2 benefits plants cultivated under drought stress, and CO2 enrichment may lead to an increase in osmotic adjustment. Elevated CO2 can also improve the water use efficiency of C3 plants because elevated CO2 causes plants to transpire less and assimilate more CO2. Elevated CO2 also affects the chemical content of C3 plants' tissues. Leaf non-structural carbohydrates, sugars, and starches per unit leaf area rise. C3 plants' concentrations of nutritionally significant minerals that affect human health like iron, zinc, calcium, potassium, magnesium, and phosphorus are declined. Effects of elevated CO2 on the morphology of C3 plants. The elevated CO2 effect is far more obvious in shoots compared to roots. The root-shoot ratios and leaf weight ratios are increased in C3 plants due to enhanced photosynthesis, cell divisions, and differentiation that led to a faster rate of leaf area growth. Elevated CO2 encouraged C3 plants to grow larger with more branches and leaves to improve the accumulations of dry matter in various plant sections. The allocations of carbohydrate under elevated CO2 conditions varies depending on species, whereby in certain species the seeds get more carbon than the leaf, shoots, or roots. For instance, elevated CO2 encourages the movement of both carbohydrates from vegetative tissues and neophytes carbohydrates to the panicle in which it is stored as starch. Effects of elevated CO2 on the yield of C3 plants. Higher yields might arise from fruit accumulating more carbohydrates. Larger fruits are a consequence of fruits accumulating more carbohydrates owing to greater photosynthetic rates in an elevated CO2 environment. Fruit weight increase in the current research at higher atmospheric CO2 levels. According to a related scientific study, tomato plants grown under greater atmospheric CO2 settings produce fruits that were heavier than tomato plants grown under normal CO2 conditions. To summarize, C3 plants cultivated in an environment with elevated CO2 produce more carbohydrates because of an increase in photosynthesis activity. Therefore, under changing climatic conditions, CO2 at increasing concentrations may lead to increased yields. C3 plant species exhibit greater rates of photosynthesis, increased growth, reduced water usage, and decreased tissue concentrations of nitrogen and protein in the presence of elevated CO2. In addition, agriculture productivity and food quality are projected to be impacted by elevated CO2 throughout the next century. Future research gains are elevated CO2 may significantly modify the nutrition, physiology, and structure of citric agricultural plants, may have an impact on the quality of food production. The strategy to ensure future global food quality of all city plants must also be included in future research that focus on the development of new techniques and the improvement of model stimulation to study the interaction of CO2 and city plants. These strategies are important to maximize the yield of city plants that can be beneficial to us.